to your second international meeting, The Future of AIT. However, I cannot be with you in Milan due to other parallel duties. And I have to admit that my voice is a little bit cracky because I still suffer from a cold. But I hope you can understand everything I want to tell you today about the genetic causes of sperm flagella defects. I have no disclosures whatsoever regarding this talk. So, talking about infertility in Germany. In Germany, we have about 6 million German women and men aged 25 to 59 years, which are unwantedly without a child. And you all know that the causes are partly on the male and the same part on the female or on both sides. But we still have about 10% where we don't know what is the actual cause. So if we have 15% infertile couples in Germany or in Europe, it would mean that at least in half of these cases, the male partner is affected, which would then result in about 7% of men which have disorders of reproductive functions. I don't think that I have to convince you that andrology is more than just counting sperm. We have a lot of diagnostic instruments in andrology, which we can use, so clinical examination and ultrasound. We can check for hormones. Of course, still the ejaculate analysis plays a very important role in the diagnostics in andrology. We have histology, but also we have genetic examination. And here I would like to talk a little bit about genetics 2.0. Over the last decades, a lot of new genes have been found and a lot of monogenic diseases are the cause for infertility. So I don't think that I have to convince you that human male infertility has a notable genetic component. For example, in non-obstructive azoospermic cases, we have identified about 20% of patients with a genetic cause. Some of them having Klinefelter syndrome, some having microdeletions on the Y chromosome or translocations or already the mentioned monogenic causes. So the majority of infertility cases in andrology are still unexplained. A recent paper by Houston et al. Have depicted the actual genes which might have a function or a dysfunction probability in andrology. This goes until fertilization. And today I would like to talk a little bit about the spermiogenesis defects and also a little bit about fertilization. So there is the postulation of an improved phenotypic classification of male infertility. And of course, there is azoospermia, which can be obstructive or non-obstructive. And we have oligozoospermia, so the reduction of the amount of spermatozoa. The more interesting ones for today are the abnormal sperm structures like lobozoospermia or these MMAF, so the flagella phenotypes. I have added those with a normal sperm count and with another genetic cause of infertility, the gut spur dysfunction. So I only will talk today about these cases here. So motility and morphology, of course, are the two essential parameters in andrology. So we know that a motile sperm is vital, it has a better DNA integrity, and it leads to higher success rates. While those sperm which are immotile or have morphological defects, they are at least potentially vital if they can't move, but they also often have a lower DNA integrity, or they might result in lower success rates. So regarding motility, this is the sperm which is bound to the surface of a glass with its head. So if you have the normal motility pattern in a normal buffer, you see it's quite equally distributed, this motility pattern. If you refuse progesterone, so the hormone which is displayed by the egg or the egg coat, so the cumulus cells, you could see that this sperm is, the movement changes. So it's going into hyperactivation mode. It's not regular anymore, as you can see now. So it is this hyperactivation mode. And you can look and visualize this mode of action of the flagellum. So this is the normal one beating pattern, and this is the beating pattern with progesterone. There are some patients where the perfusion with progesterone does not lead to this hyperactivation. So in these patients, the amount of sperm which do hyperactivate is extremely low. So the cause of this is well known. It's the Sperm channel, which is only located in the tail of the spermatozoa. So the Sperm channel, if it's intact, of course, normal fertilization can take place because the sperm can act in its hyperactivated mode. But if the Sperm defect is the Sperm channel is defect, then there is no fertilization possible. And indeed, there is a test now available, which my colleagues developed, and we did it like that. Every normospermic patient entering our laboratory had to undergo this test. And you see here in green that most of the spermatozoa were normal in this Sperm activity test. However, there were a few patients where these tests did exhibit differently. So these were not active anymore. And we could identify among these 9 patients which were definitely in the end had a Sperm activity test which was not normal. We did a genetic analysis and we found that the Sperm2 gene was deleted in at least 8 of these 9 patients, leading to inactivation of the Sperm channel. And the point is that if you do with these kind of patients an IVF, it will always fail because they cannot activate the sperm and do not use the hyperactivation mode to fertilize the egg. So in these cases, only ICSI is the possible method of choice. Another very interesting issue we found in these patients is that they not only had the Sperm2 gene deletion, but also next to it the gene deletion of this STRC gene. Not all, but some of them. An STRC deletion leads to a mild to moderate hearing impairment. And it's also called the deafness infertility syndrome. These patients had some hearing disabilities, but it was never found out why they had this. And now they could at least, now they have a diagnosis. They know that it's not only the Sperm2 gene, but also this STRC gene, which leads to their hearing impairment. So here I have just 2 small quick case reports with the Sperm2 channel, Sperm2 protein deletion. So this is one patient, and you see normal concentration, normal total sperm count, normal motility. They did, before they came to us, 2 inseminations externally and had no success. Then by chance, they came to us and we detected this Sperm defect in 2019. And 1 year later, that woman, she was already 37 years old. We stimulated her and she had a high number of eggs retrieved, 20 in total. And due to insurance problems, because the insurance would not pay an ICSI because the spermiogram was normal, we divided the eggs into 5 with IVF and 15 ICSI on our own costs. And of course, it happened as it was proposed, no fertilization in the IVF. So this is an egg after IVF, and you see that all the sperm are bound to the zona pellucida, but none of them made it into the egg. But in ICSI, we had 9 of 15 fertilized just normally. Unfortunately, we had no pregnancy in that trial. The second case, again, you see normal sperm count, normal motility. In 2017, they came for the first time to us and did some 2 times timed intercourse and had no success. In 2020, some years actually later, they then thought, okay, we have to do something and go on now with IUI or IVF. And by chance, we detected again this Sperm defect with our Sperm activity test. 1 year later, she started with ICSI, and she was already 39 years old now. And that is exactly the problem we encountered, because we had either no response or only one egg, no egg. And finally, in the 5th attempt, we had 2 eggs, 1 fertilized, and she got a single embryo on day 3. And luckily, a baby girl was born in November 2022. So coming to the other point, now the morphological alterations. There are, of course, the sperm which are non-motile but vital, and there are those sperm which are morphologically impaired. There are several variants. There is, for example, the Flagella defects. You see these sperm look like with broken tails. Then you have the globosospermic patients, where the sperm have very round heads, and that often is a cause of a homozygous deletion of this DPY19L2 gene. And there are also rare cases of macrosospermia. Here, there is a homozygous frameshift variant in the RXC gene. And the interesting thing here is that these sperm are always tetraploid, at least tetraploid. So you see that they are melted together and they have 2 to 4 sperm tails and a very large head. In these cases, no ICSI is possible. There is no treatment possible to get sperm with a normal chromosomal content. In case of globosospermia, you can use the calcium ionophore to directly activate the egg, and that is potentially helpful. And the third ones is MMAF phenotypes. I would like to go into detail now. So these sperm here, control sperm, have very faint and thin tails. They are shortened, or they are look like this here, or a little bit thickened. And also here it looks as if there is a little tail which is curled at the end of, or directly behind the head. And this phenotype is called MMAF, multiple morphological abnormalities of the flagellum. The flagellum is nearly the same like all cilia in our body. So they are microtubule-based ultrastructure with 9+2 axoneme in the middle. So 9 on the outside and a central pair. Plus some Dinian arms, these are the gray structures, the axonemal ruler in between here, depicted also here, and these radial spokes, which look like nil mushrooms. So this is how a normal sperm should look like. Here you have the 9+2 axoneme picture, but there are also sperm. In this case, they are mutations in the ruler complex, and you see that the normal 9+2 appearance is not anymore there, and also the head and the mid piece look very strange. This is another example here. It's in the radial spoke, and you see that there are no connections to the central pair. And again here you see that the mid piece and then later the tail is affected here. These patients with these celiac syndromes, they often suffer from primary ciliary dyskinesia. It is a rare genetic disorder, and it affects all motile cilia, which we have in our body. And we have a lot of cilia in our body. There are many in the eye. There are some in the heart. We also know, of course, there are some in the oviduct. We have some in the brain, in the respiratory system, and also in the kidney, and lately, as male, also in the sperm flagellum. So what we did in Münster, we teamed up in the Clinic for Pediatrics and Adolescent Medicine, which have a lot of PCD patients, young patients, children, together with the medical genetics and our center and Professor Stümker from the physiology. We now look and analyze these cases. So here are some of our patients with have, for example, problems or mutations in the ruler complex, and you see that these tails are swollen or curled at the end in different it looks different in every sperm. And this is one where the central pair is affected, and these really look as if the tail is completely curled directly behind the head and not cannot be identified in detail. From literature, it is known that some of these genes, here is a CFAP44, 43 mutations, that these patients have the chance to become father and mothers. However, it is quite a task because these patients often have no motile sperm and often also no normal morphology. This is one patient, he was 28 years old, his female 27, and he had DZIP1 mutation. DZIP1 is a DAS interaction protein and is associated with the centrosome in the midpiece of the sperm. As depicted earlier, they look as if the midpiece and the tail is really curled behind the head. So what kind of possibilities do we have? So there are several vitality tests for sperm in case the sperm are immotile. For example, can we use the Hoss test, the hyperosmotic swelling test, where sperm is gently placed into a hyperosmotic solution, and in case it swells, you know that sperm is vital. You can use activating substances to at least try to identify those which have a little tail twitching, and then you know that that sperm is motile. Mechanical techniques, I'm a little bit more reluctant. They are very difficult in identifying motile sperm. And lastly, there is also the possibility to use a laser and have a little shot on the tail, on the tip of the tail of the sperm. And by seeing this little curl, a little bit like the Hoss test, you can identify sperm which are motile, but not, which are non-motile but vital. So here's the film. I'm very sorry, it's enlarged from our camera. So here is the impact of the laser, and if you look directly here on the tip, you see that there is a little curling. I will play it again. Again, here is the laser shot, and you see this little curling of the tail. That means this sperm is vital, but just cannot move, and you can use it very good for ICSI. So what did we find in these patients on the day of ICSI? Well, we found exactly those described in the literature with those little curls in the, behind the head. But we were also lucky. We found some sperm with tails about 1/3 length. And we also found one DD tested with the laser and even one A sperm with a normal tail. And altogether, it took us 1 hour to have this 1A, 1D with a laser normal, together with 6 sperm exhibiting this 1/3 sperm, where the laser actually did not work that well because they were very straight and very thick, these tails. And we had 13 of these very small tailed sperm where no laser test was possible. We retrieved 20X and 14 were in the metaphase 2 stage and were ejected with these kind of sperm. We did add calcium ionophore for egg activation because it is known that these sperm have problems with the activation and the calcium signaling in the egg themselves. So it might be a good idea to add calcium ionophore for egg activation. In the end, only 2 of the 14 eggs were fertilized. And interestingly, only from those sperm with the 1/3 tail, not the A with the normal tail, not the D with the normal tail that work. We unfortunately had to do a freeze-all. In Germany, we do mostly pronucleus or later blastocyst stage freezing due to the risk of OHSS. So there was a frozen embryo transfer cycle planned some weeks later, and that patient insisted to have a day 3 transfer with both PN developing to a 6 and to a 7 cell stage. And she was lucky. A positive pregnancy test later on was seen, and also the heartbeat of just 1 baby. And there was the birth of a healthy baby girl on the 21st of January last year. And they are coming now for the next ICSI, and now we know that we rather retrieve those sperm with the 1/3 tail to be successful a second time. So I hope I could convince you that laboratory techniques, for example, in case of immotility of spermatozoa, can be used to identify vital ones, for example, the laser test or the modified Hoss test. Moreover, the use of a calcium ionophore might in some cases be beneficial. The Sperm activity test clearly helped us to identify loss of function of the channel in some of these patients, and by that circumvent, for the new patients, to do IUI or IVF, because that will never work. And overall, there is definitely a notable genetic component in human male infertility, and the genetic causes of these Flagella phenotypes can be identified quite often in these cases. But of course, there are other genetic causes that might still be unknown and difficult to identify. But due to the genetics 2.0, I'm pretty sure that more genes will be detected and these patients will get a cause for their infertility. This is, of course, not a one-man show. There are many, many people involved. And I would like to say thank you to all those which support us in the IVF lab from either side, from the gynecology department and from the andrology department. And with this, I would like to thank you for your attention.